So this video is going to give you an overview of the sorts of genes and mutations that are involved in the generation of cancers. So cancer is fundamentally a disease of unregulated cell division. So that's why you get tumours, is basically because cell division gets out of control, it's not regulated properly, so you end up with these masses of cells, and then if those masses form in a particular organ, so let's say it forms in the kidney, and then that affects the kidney function, so then you end up with the pathology of the disease. So if we're going to understand cancer, then we really need to understand cell division. So there's another video about the cell cycle that you might find useful for this, that goes into the cell cycle in a lot more detail. Today, this video is just going to be an overview of the cell cycle, but there is that one to have a look at if you want to. So let's consider for this one the difference between two different states. So we can have the healthy state and the cancerous state. Okay, so we need to think about what's different between the two of them. Okay, so just as an overview of the cell cycle, so you might remember we have the cell cycle, so we've got G1, S, G2 and M. And as I say, there's a whole other video on the mechanisms behind the cell cycle if you want to go and understand that. But there's these other states that we can have where we can go from G1 into the G0 state. So the G0 state is not dividing. Okay, so we can either be in the cell cycle or we can be in a G0 state. So what I'm going to think about in terms of a healthy G0 cell. Okay, so actually in a G0 cell, we're in this G0 state. We're not going into the cell cycle. We've exited the cell cycle, so we're not dividing. So we've got no division happening. What happens in a cancerous state is we have our G0 cells, and for a variety of molecular reasons, so usually we should go out of the cell cycle and into G0, but in a cancerous situation, what we end up doing is going back into the cell cycle, and we end up having unregulated cell division. So in the healthy cells, so in your skin or in your liver or whatever, most of your cells will be G0 cells, so they're not dividing. You're not going into the cell cycle, so actually this bit isn't happening. You're in the G0 state. Whereas in a cancerous cell, your G0 cells have decided that they are going to go into the cell cycle, so the cell cycle starts when it shouldn't do, and that's what gives you cancer. Okay, so cancer is fundamentally a disease of mutation. So we have various different mutations that take us from one state to the other. So, and kind of the more mutations you accumulate, the more likely you are to go from your healthy G0 to your cancerous situation. So, so things that cause mutations, and other than just age, we might have carcinogens. So, for example, in tobacco smoke, there are compounds that actually bind into your DNA. They shove themselves into the DNA and cause mutations. So, tobacco smoke is cancerous. UV damage. So, if you spend a lot of time on sunbeds or in the sun, you're more likely to have mutations. So, you're more likely to go from the healthy state to the cancerous state. Okay, so. But the question is, what are the mutations? What genes are we talking about to get us from this healthy state to a cancerous state? And there are two types of mutations. So one of them are mutations that reactivate going into the cell cycle. So we call those oncogenic. mutations. So these mutations are, if we think of an analogy of a car, and is a car going to crash? Oncogenic mutations would be the accelerator is jammed on. Okay, so the accelerator is jammed on, so you're more likely to go from G0 into G1. So there are a bunch of genes that in the unmutated state we call proto-oncogenes. So if they're working as they should, they're not cancerous. So that would be that the accelerator is working correctly. But if we have a mutation as a result of DNA damage, then those genes there, if they get switched on by mistake, then those are oncogenic mutations, so the accelerator is jammed on. So examples of oncogenic mutations are things like, there's one called RAS, there's one called EGFR, which is the epidermal growth factor receptor. There's one called MYC. These are all genes that are in the growth factor signaling pathway. So there's another video on how that pathway works. But there is a pathway to get you from G0 into G1, which is usually switched off. Usually you want to stay in G0. But if that pathway gets mutated, then we go from G0 into G1. So we have these oncogenic mutations. So most of those mutations are mutations in a growth factor signaling pathway. Okay, so there's another video if you want to go and check that out. Okay. So oncogenic mutations are as if your accelerator has been jammed on. Okay, but in a car, if your accelerator is jammed on, well, you still won't crash because you've got brakes. Your brakes are working. And the cell cycle has brakes as well. So the cell cycle has brakes that we refer to as, let me just change my pen colour. We have genes that are called tumour suppressor genes. Okay, so these are effectively your brakes. So in the healthy version, your brakes are working. And what your tumour suppressor genes do is to repress the cell cycle. Okay, so usually in a healthy G0, in addition to not activating entry, we've also got tumour suppressor genes which are switching off the cell cycle. Okay, but in the cancerous state, those tumour suppressor genes that would normally be repressing, they get mutated too. Okay, so that's the equivalent of your brakes are not working. So now you're in a really dangerous situation. So in the healthy cell, you've got two different things. So your accelerator is working properly and your brakes are working. You've got two different mechanisms. If one of them fails, you might be okay. But if both of them fail, you might be in real problems. So if your accelerator is jammed on and your brakes have failed, then you're very likely to crash your car. So in this analogy, if your tumour suppressor genes are mutated and you've got oncogenic mutations, then it's very likely that you will get into the cancerous state because all of the controls on the cell cycle have been switched off. Okay, so tumour suppressor genes. So we named some oncogenes over there. Tumour suppressors, we've got things like, and the classic tumour suppressor gene is a gene called P53. And P53 regulates all sorts of stuff. So it regulates apoptosis, it regulates DNA repair, it regulates the cell cycle directly. So P53 is mutated in about 50% of cancers. So there's a lot of work happens on p53 and how it controls all sorts of processes. There's also a gene called RB, which is retinoblastoma, which was identified from eye cancers. And then there's the gene BRCA2 or BRCA2, which was identified in breast cancers. So these are all tumour suppressor genes. So normally their job is to switch off the cell cycle, but if those get switched off and we have oncogenic mutations, so the G0 to G1 pathway gets switched on when it shouldn't do, then we are more likely to get cancer. So kind of the more mutations that you have, the more different pathways that might be hit, but also you might have going from a heterozygous to a homozygous situation. So if we think about having some chromosomes over here, so we could imagine in your healthy version you've got your chromosome from your dad, chromosome from your mum. Okay, so the healthy version would be that neither copy is mutated. We could have someone who is predisposed to developing cancer. So they are born or have already accumulated a mutation in one of their copies of a tumour suppressor. So let's say it's p53. So if you're born with one copy of p53 being faulty, you're at a higher risk of developing cancer. If you then accumulate another mutation, so now we've got both your chromosomes, both of them have got faulty p53. Now you've got a really quite high risk of getting cancer. Your both copies of p53 are not working. So if you become homozygous for a tumour suppressor, you are really at quite high risk of generating cancer because your p53 doesn't work anymore. So in a lot of genetic screening and sort of personalised medicine, one of the things we're looking for is what's happening. Have you got predisposition to cancer? So of these tumour suppressor genes or these oncogenic mutations, we know quite a lot about them. So we can do genetic testing and genetic screening to work out whether you're in a healthy situation or whether you've got a predisposition towards cancer. So particularly with BRCA2, which is associated with breast cancers and a lot of female cancers, it's looking at, have you already got one mutated copy of BRCA2? That runs in families. So if you've already got one copy of BRCA2, you've got higher risk of developing cancer. And patients are increasingly using those genetic tests to make decisions for themselves about preventative treatment. So it might even be as extreme as a preventative mastectomy, because we've managed to look at the genome and find out what your genetic predisposition to cancer would be. But it's all about these two systems. We've got our tumour suppressor genes, which usually repress the cell cycle, and we've got our oncogenic mutations, which get us from G0 into G1. And you have to have both of those systems not working properly in order to get cancer, and that is fundamentally accelerated by mutation.